Well, today we are going to learn more about testing our biological age and how we can see how fast or how slow we're aging with Ryan Smith. And this is kind of important to me because then you can see how fast, at least I can see how fast I'm heading to my death. So I can be sure that what I do is that's necessary to make this journey slow and painless and enjoyable as possible. So really, it's important for me. And Ryan's going to clear out a lot of confusion and all these testing kits that are on the market that measure all kinds of different things in our bodies to tell us how old we truly are and not just the number of candles on our birthday cake. And Ryan Smith, to give you a background, he's a biochemist and co-founder of a company that helps us figure this all out. And it's called TruDiagnostic. And TruDiagnostic has been circulating in the biohacking community for a while now. Everybody's talking about it. So I'm really excited to have him on and explain this to you. And he's been on many podcasts before with some of the biggest names in health and wellness. And all of my biohacking besties and doctors I know personally have given rave reviews on both his incredible knowledge and experience, but the product as well. So a little bit more background. Ryan decided to open up a pharmacy in the US after he graduated with his biochemistry degree. And what I love is that he focused on peptides, which you all know I am a fan of, and general pharmaceutical drugs. And his pharmacy, called Tailor-Made Compounding, became the fourth fastest growing company in healthcare in the US, which he later left in 2020 to create TruDiagnostic, so he can help people prevent the diseases of aging and not just managing them. And so this is both a lab and a health data company that is doing an amazing job so far at supporting our doctors in the functional medicine space, including my own. And I'm not sure about the other companies out there, but TruDiagnostic is collaborating with over 30 clinical research studies that look at which interventions work best for health and longevity. And they've already tested over 13,000 patients and have one of the largest health databases in the world. And it's pretty amazing what he's done in just 2 years. I am impressed. So let's finally meet Ryan Smith. Welcome.
Yeah, thanks so much for having me, Zora. It's great to be here, and I appreciate the introduction.
Yeah, did I get everything right? Probably have more than 13,000 people by now.
We do. We have a slightly more, but yeah, we keep growing, which is great because as we get that more data, we sort of learn how to use it a little bit better. So that scale is slowly getting up there, and as a result, we're increasing what we can find on some of these reportings.
Oh, that's better for all of us. I want to first explain what is biological age, because some people may be confused between biological and chronological age.
Yeah, definitely. And it's sometimes a difficult question to answer, but generally this idea of phenotypic variation is where it starts. And phenotypic variation is sort of the difference in aging across people of even the same chronological age. So for instance, we all know people in their 75, 80s who look like they're in their 50s, and then vice versa. We know those people are probably aging a little bit prematurely. And so that difference is can be really difficult to conceptualize, but it's a big issue because already chronological age, or the number of sort of candles on your birthday cake, is the biggest risk factor for almost every chronic disease and death. So we know the importance of age, but this difference in the phenotypes of age, how people look as they age, is important to capture as well. Because if we can really capture how people are aging from the inside, or their biological age, we can really get a better idea of how well their body is aging and how we can either slow that process or do things to prevent that process.
I think it's super interesting because we do all see those differences. And as a gerontologist, I sometimes wonder how biological age, if that would ever be considered when it comes to healthcare rationing, right? Some people are denied healthcare because they're too old and they're going to die anyway, but is they want, what if you do have a chance, right? And you say, come up with your certificate. I actually have a biological age of a younger person, right? How do you think this biological age will affect healthcare or in doctors in
Yeah,
general?
so yeah, that matter of triage is an interesting one ethically that I haven't really thought about. I think that already certain aspects of age are really important to consider for even societal funding. How are maybe underprivileged groups having accelerated aging patterns as a result of their demographics or environment? So I think there's a lot of ethical questions which can be asked there. But I think fundamentally, what we really want to look at is this is the number one biggest risk factor, something we know impacts everyone to a large degree, impacts almost every disease. And as a result, something we want to measure, so we can then, find out what works to reverse it. I think that's the first step. I think once we start to know about what reverses it, we can really consider some of those grand, ethical questions about implementation of this. And it's already starting to happen, as I mentioned, with particularly younger people and societal and economic related disadvantages. But at the very least, what we want to do first is find the best signal for biological age, and then the things we know that can reverse or change that signal.
So yeah, you talk about signals, and that kind of leads me into the next question, because there's so many kits on the market, and what signals are we looking for? Because there's glycans and there's the telomere testing, and it can be really confusing. And everybody's, even my scale gives me a body age. Like, how accurate are all of these tests, and what's the difference between them, and how can somebody know which one to even choose?
Yeah, great question. One that, again, is a little difficult to answer because whenever we talk about accuracy of these tests, what we're really talking about is it able to capture the aging process? And so there are a couple categories of things there that we really wanted to do. One is it must be able to sort of capture where someone is among their aging journey. And because it's the biggest risk factor for chronic disease and death, we needed to have very high hazard ratios or predictive sort of associations with those diseases. So those are really how we would calculate accuracy in these biological age fields. It needs to be unrelated to a disease. It needs to be something that can be tested repeatedly. It can work in laboratory animals, and something that is highly predictive of these negative outcomes we know are associated with the aging process. And so those are a lot of categories. Those are sort of defined by the American Federation of Aging Research. And so really these biological age markers have really taken a big step forward really recently because of sort of this sort of the advent of this multiomics, where we're getting very, very, very good at seeing things on a molecular scale. And so really the reason we would say epigenetic methylation is the best diagnostic for age is because of that hazard ratio, which is incredibly, incredibly predictive. So generally, if you're aging quicker than your chronological age, that means you're at an increased risk. And then vice versa, if you're younger biologically than your chronological age, that means you're at decreased risk. And comparatively to any of the other platforms, if you're looking at even just blood-based biomarkers, or if you're looking at glycans or even proteomic clocks, the one that is by far the best and has been stated as the best now in several review studies tends to be this epigenetic methylation based clocks. But it gets even complicated there because that clock is becoming even better and better and better. And so what I would say is that in the future, methylation clocks won't necessarily be the best clock. You'll have these composite biomarkers that will look at everything which is age associated in humans, everything from metabolomics, proteomics, transcriptomics, epigenetics, and these sort of wide array clocks that capture a lot of different things in the body. Those composite clocks will be the best. But right now, I would say that epigenetic methylation has been the thing that is published with the highest hazard ratio, meaning it's the most predictive of those age-related diseases.
Okay, now you have to explain, what is epigenetic methylation?
Yeah, definitely. And this one's easy to answer because it's essentially the on or off switches on our DNA. I always like to describe to everyone, if you took any cell in your body for DNA sequencing, if you took your skin cells or your hair or your saliva, that all those cells have the same baseline DNA. But they obviously behave very, very differently. There's a reason that your skin behaves like skin, your heart behaves like a heart, and that's by what genes are up or down regulated for the expression of that type of tissue. And that's regulated by these markers or epigenetics or above the genome, as it sort of translates out. And those are basically markers which can control which genes are turned on and turned off, and then ultimately control how that cell behaves. And so that's sort of what we're looking at is epigenetic methylation, which is sort of the off switch, essentially. Whenever epigenetic methylation is there, generally you have less transcriptional activity, which means you're making less mRNA, which leads to less proteins and peptides, and that central dogma of biochemistry. So we're looking at the off switches and sort of these methylation groups, which are attached to certain locations in the DNA. And methylation itself is not good or bad, but these patterns happen in tissues, and they sort of happen with age. And that correlation with age was really that big discovery, which happened in 2013 by Dr. Steve Horvath. He created sort of this first pan tissue clock, which could really tell us how old each cell or tissue was. And that was a big step forward in sort of this area of biological aging.
Yeah, I like to explain, people, when we think about behavior and genetics, in turning switches on and off, the way I understand it is that we want certain genes on and we want certain genes off. It's like you never want them all on or all off. It just depends if it's a cancer cell or if it's, some anti-inflammatory, marker. So we need to Yeah, I think understand a little bit. Yeah, epigenetics is the behavior of our cells and our genetics, and we just need it to be in optimized in order to try to lower our risk of disease. And this is what you're looking at, or through TruDiagnostic, that's the marker you're looking at. And so when I think about, when we go through the kit, so I received my kit, I opened it up, and then there was a questionnaire that I had to fill out. I had to pick my finger, draw blood, put it into a tube. Although I think now you're having blood spotting paper coming up, which will be much easier. Seal it, send it back, and then I get my results in what, 4 to 6 weeks or so, right?
Usually should be right around from 2 weeks from the time we receive it. So, maybe 3 weeks generally is,
Okay.
I would say that from start to finish, that turnaround time. But yeah, that's exactly the process. And we are using those blood spot cards now, which is a little bit easier for collection.
So then, but the blood is coming from my finger. Now, is that enough accuracy in terms of finding out my epigenetic methylation? Because what if I pulled it from my toe or from my elbow, or does it differ from different places that I take the blood from?
So not necessarily, venous or capillary blood is generally going to have the same makeup of cells, which are generally, those red blood cells that we find as the majority of blood are actually do have no DNA. So what we're looking at are these leukocytes, these sort of, your B cells, your natural killer cells, all those immune system cells, and quantifying that. And that's generally the same wherever we would find it in the body. And so, but that's, very important to mention is that those immune cells, as all are different epigenetic expression, B cells and T cells have different epigenetic markers. And so what tissue type you're measuring is very, very important for this type of investigation. We use blood because that is by far the most established tissue type from almost all of these algorithms, and what most of the literature is studying sort of going forward. So even just with one test on methylation, we can actually probably give you a lot of different outputs, all the ones we're doing for aging, but more we can even do for general health, and in a lot of different and specific categories.
Yeah, no, it makes me to think about, so how different then is measuring your DNA methylation than the glycans, for example, because or telomeres, because we're talking, you said superior, okay, we got that, but by how much? I mean, is it really big difference?
Significantly bigger difference. And there's a great study, which Zora, I'll try and send to you, and maybe we can post with this podcast. It's sort of a review of these biological age markers from 2017, which is a little bit outdated now. Even these methylation clocks are getting even better, but it sort of talks and balances this sort of the number of studies published versus the hazard ratios for these things. And generally these hazard ratios for these epigenetic clocks, particularly the second and third generation clocks, which we might want to go over, but are much, much, much more predictive. We're talking, increasing prediction rates by 20 or 30%, which are very, very significant.
Oh, okay. So then, because yeah, there is a difference. So I just want to get people to understand the difference. And 23andMe, for example, these DNA testing, you spit in a tube and you send it out. So they're collecting a different medium. And how would that differ from TruDiagnostic?
Yeah, so both in collection method and in what we're looking at are going to be what differs there. They're using saliva, which has a lot of those same lymphocyte cells that we talked about in blood, but it also adds those epithelial cells, and that can really throw off some of how these algorithms were trained, especially if they were trained only by collecting blood. And so that is one of the biggest things you want to look for in any company who's looking at your epigenetic biological age. You want to make sure that the algorithms that they're using, first off, are published, and that way you can really trust their results and how they relate to disease. The second thing you want to look at is making sure that that published algorithm has been created and used in the same type of tissue type as the one that you're collecting. I always use this example, but if we were to take those, use that 2013 Horvath clock, the one that first came out, and we were to measure brain tissue, we get much, much lower ages than if we looked at blood. And if we measured your breast tissue, we get much, much higher ages than if we looked at your blood. And so in order to be accurate, it needs to be sort of the standard method. And so we're still using blood for that. And that's what I would highly recommend for anyone.
What's interesting, you mentioned the algorithms being published or not. So you're saying that other kits out there are not publishing their algorithms?
Yeah, it's a little bit of a strange field because the algorithm or the IP behind those predictive qualities, right? The information that we're getting is pretty much the same information that a lot of researchers might get. It's the same that a lot of commercial companies might get, but it's the interpretation of those algorithms which have been created, which are really the IP. And so a lot of companies, not necessarily TruDiagnostic, but a lot of companies don't want to release that information because then they're giving away their insights. And so it's one of those double-edged swords where you either have to give it away or you have to let people trust you, one of the two. And
Mhm.
so we have definitely taken a position to publish and as much as possible, because that's how we're able to link these algorithms to their ability to predict disease. And that's really why this is important, so that we know that if you have accelerated aging, we can accurately identify that and accurately identify how it's increasing your risk for all these chronic diseases and death. We believe that this should be a risk factor that can be mitigated and tracked across a lifetime. And so in order to do that, we think you can't just take our word for it. You really need to see that we know this works through large population studies.
Wow. So you're being super transparent, but at the same time, you're risking somebody copying your algorithms, right?
Correct.
And so, yeah, that's
Yeah.
you're
It's tough.
taking a hit for the team.
Yeah, but I think it's better for the industry. I think that, as you mentioned, these ideas of biological age, there's a million options to choose from. And so I really think that if you're going to be using an option, you need to know that it's the most accurate option to give you the most information. I think hopefully we can do that.
Hmm. So you give a lot of reports. I've done other kits and it's quite simple and a couple of pages and you just kind of get it and you just like, want to know, okay, I'm 36 or whatever. But your reports are like an encyclopedia, and you also have reports that you send to the doctors as well, because they get some of their information. What are the reports that you're offering, and what do we see that's especially and which ones are different than the others that are on the market?
Yeah. So whenever we first started, we just had a biological age report, and that was one that was meant to tell you how old your body is, at the very baseline epigenetic level. And so that was the first report we started with. But as we started to get more patients, we always talk about our mission here is to sort of create the Rosetta Stone of how to interpret these methylation marks. So we know exactly how to read them and know exactly what it's telling us about patient health. And as the years have gone by, epigenetic methylation has been rapidly progressing. It's been published, published studies are happening every week on how to interpret this. And so we generally will update some of our algorithms and reports fairly frequently. And so from an aging perspective, now we do much more than just your biological age, but we also do things like your immune system age or your immune cell subsets. So how many CD4 or CD30, how many CD4 T cells do you have? So how many CD8 T cells do you have? Natural killer cells, basophils, lymphocyte counts. That way you can get an idea of how your immune system is aging and how well it's doing right now. And so we do stuff like that. We do things like telomere length. Telomere length has been one of those markers of biological age for a long time, but it's not been that predictive. It has a really low hazard ratio, usually a 0.002 hazard ratio, just to give you an idea. So 0.002% increases, right? And so, or so I should say 0.2% increases. And so that's really not that predictive of these health outcomes, but it's still an important part of aging. So we're able to do that. We also do this pace of aging. So not what is your overall biological age, but right now at this very moment, what's your speedometer of aging? How many biological years per year are you aging? That's definitely our favorite algorithm, sort of the first ever third generation algorithm that was created with Duke and Columbia. And so that one's by far our favorite. It's one we always recommend to prioritize. And it really tells you how you're doing now versus how your body is doing overall, which, sometimes is very important as people have maybe lived, some unhealthy lives in the past and then now are living a little bit better. And so it gives us a little bit more of a temporal idea of how you're doing. And if you're doing well, that's great. That means you're probably going to improve your other markers over time. But in addition to that, we have disease related markers, things like, can we predict your risk of diabetes, which we can up to 7 years earlier than fasting insulin and A1C. You can predict diabetes. We can tell you if you're likely to lose weight with caloric restriction. We can tell you how much you've smoked across an entire lifetime. If you're a current, never, or former smoker, we can tell you how much you're currently drinking.
how,
And
how can you
that's just
do that? That's that how is that you can look into the past? I'm sorry to interrupt you, but I just like I've
yeah.
heard other people tell me their results like that as well. They can't hide it because you do have this questionnaire, and I'm part of me kind of goes, wait, okay, how much are you taking from what I'm telling you, and how much are you actually seeing? And what if I didn't? It's just like, what if I left the questionnaire blank? What would the results show?
The results would not change at all. We do not use any of those survey responses in order to generate any of our metrics. The only reason we do that is because context, we feel, is very, very important. Most people don't know what their C-reactive protein should be or their triglyceride number should be, but everyone knows what their age should be. And so we want to make sure that people aren't scared of this metric. And in order to do that, we want to report on the thing that we know changes these things back. And so we ask a lot of questions, for instance, about things like sleep or early childhood behavior, things we know with high epidemiological correlations to increased or decreased age. So we can tell you maybe not just what your age is, but maybe give you context for why your age is that way too. And we think that's very, very important because we don't want anyone to be scared of this metric. You can always take this into your hands. You can reverse it. And so we want people to approach this with no fear, and to really to go into it as level-headed as possible with all the information they can. And so if we didn't answer any of those registration questions, it wouldn't change any of our algorithms.
Okay, so now I'm wondering, yeah, I have quite some questions that I'm not even sure about because I sent in my report and it's supposed to be ready any moment. But what I didn't get a chance, and I'm sure people are wondering about this as well, is when I get my report, will I be able to read it all by myself, or do you give a consultation, or do I take it to my doctor? What do I do?
So the major people that we generally work with are physicians, and so that's where the majority of reports are going. But in a direct-to-consumer fashion, a lot of those same physicians we work with offer to do consults with our patients to give a little bit of an introduction to the process. And so you can absolutely have a consult with one of our participating physicians. So hopefully, we'll give you a little bit more context, or you can ask us questions even directly, and we're happy to help. But the reports are geared to be pretty self-explanatory. They talk about how these algorithms were created, which is one of the most important things. They talk about how they are linked to different health outcomes, which again is one of those most important things as you think about the application. And so hopefully they're pretty self-sufficient. You can read through them themselves. It is a good bit of information, but we can always sort of narrow down and drill into the things which matter most. And I think that rate of aging is definitely one of those things.
Mm. So you said the algorithm is continually changing. So if in my report it says I'm 42 instead of 52, then you change the algorithm and says, oh, we made a mistake. You're actually 50 or younger. Does that happen? Do you inform us?
So absolutely, and generally we won't change the algorithm, but these algorithms are becoming better. And so generally, if we do add something, it's usually a new algorithm, which means we wouldn't take away any of the old algorithms. We would just generally add more information, and that sort of goes into how these algorithms have progressively gotten better, even over the last 2 years. They've gotten significantly better, and generally at first these algorithms were trained to predict someone's chronological age. So the first examples of this algorithm were not even used in health. They were used for things like forensic crime scene investigations to see how old someone was if they left their DNA at a crime scene. They were also used to date refugees in Europe to see if they were, minors or adults, and then therefore eligible for asylum. And so, those were the first applications. But then these clocks became, I would say, geared toward health, where they saw that the differences, the delta between chronological and biological age, was very, very predictive of health outcomes. So I always like to give an example in the case of cancer. For anyone who is 1 year older biologically than chronologically, they would increase their risk of cancer over the next 3 years by 6%, and they'd increase the risk of dying of cancer over the next 5 years by 17%. And so you can sort of see how you can do that math, where someone who's 2 or 3 years older would have a significantly higher risk of developing or dying of cancer. But you can do that with every disease, right? Objectively, you can do that with all of these diseases, which is why this is so exciting. One marker to really look at the biggest risk factor for almost every disease. And so that's what we really like to use it as and look for it in. But these clocks got better because instead of being trained to chronological age, right, the better that those clocks got at predicting chronological age, the closer they got to something we could just ask for, right, which is your birthday. And so instead, what we have sort of changed to is how do we best detect this sort of system biology of those markers of aging. So we don't want to measure your chronological age. We want to measure how your body is progressively losing the function as you get older. And so in order to do that, those second and third generations have used a lot of blood-based biomarkers, things like, for instance, telomere length, body mass index, and triglycerides, lung functioning measurements. Those are better captures of this aging process than just the chronological age of an individual. And so now that's how these algorithms have been trained, and they continue to be updated with more and more information, which makes them we can continually get more accurate. And so that's where we're at now.
Mm. So what I thought was really interesting, I heard from some of my clients, I know they're doing TruDiagnostic, and they said this pace of aging, this rate of aging report is super cool. And I saw there was a turtle and a rabbit, right? There's sort of this speedometer, and you want to be in the turtle, right? You want to be closer to the turtle and aging slower than the rabbit. So that was one. I think you mentioned the immunity report was one of the most important. People enjoy the biological and the pace of aging report. What other reports are you seeing that people value?
Yeah, so I want to just place special emphasis on this rate of aging report because it is, as I mentioned, that first third generation algorithm, and therefore one of the most accurate, the most predictive disease. This is a really interesting report, which is because it was sort of gathered longitudinally. All these data sets, instead of looking at a lot of different people over a lot of different age groups, this algorithm looked at the same individuals across their lifespan. And so this really started in New Zealand in 1972. And they really followed a group of children from the time they were born until all the way until 2019, where they were age 45. And so we're actually able to follow the same individuals over the course of their lifetime and able to use a lot of the biomarkers we took for them to generate this instantaneous pace of aging. And so that's a 19 blood related biomarker score. It includes some of those things, as I mentioned, like leukocyte telomere length, like lung functioning spirometry metrics, like physical function metrics, even brain MRIs and facial aging pictures we were able to take. And so what we saw is that at age 45, all of the quality of life metrics. And then I think this is important too, because when people think about longevity, they oftentimes think about how long they'll live. They don't think about the quality of life that they'll live and the improvements we'll see there too. But we saw that generally the faster the rate of aging, the worse your performance on balance testing, the worse your performance on IQ testing, the worse your mental processing speeds, your grip strength and muscle mass, and especially even your facial aging. All of those things decrease as your rate of aging gets faster. And so what we really want to do is to make sure that as much as possible, we keep that rate of aging as low as possible for as long as we can. And so that's probably our favorite. It's also responds to classical interventions that we know work to decrease aging with like caloric restriction, for instance. And so it is by far our favorite algorithm. That's one we always recommend to be prioritized as the number one algorithm. But we also, as I mentioned, do some of those things like telomere length and those other immune or biological age reports. And it just gives us a really good picture of aging. You know, what for you is aging quickest? Is it your immune system? Is it your overall aging? Is it your instantaneous pace? Are you doing something now that we really need to change? Or is it telomeres? You know, and are we seeing, you know, things like maybe high markers of senescence? And those are some of the reports we're going to come out with in the future. Senescence-based report, inflammatory-based reports, where we can look at IL-6 and see reactive protein and TNF alpha, a lot of those inflammation reports. And so we'll even be able to report and predict your things like your VO2 max or your grip strength or your walking speed very, very closely here in the future.
Oh, wow. Interesting. So more interesting reports to come. So you'll be keep adding to that. So if somebody does a report or they do their test, they just do it once, or do they repeat?
Yeah, so we will generally give updates to your data for as long as we can. And we're using that sort of classical measurement. We use what we call an EPIC 850K array, which is sort of the standard of all clinical research now in the field of epigenetics. So as researchers from any institution come and publish information about what they found with these associations with age, we'll be able to update our report. And we'll continue to do that for many, many years to come. And so hopefully you'll be able to continue to use that same time point for a lot of information. But again, these things change. And so depending on how you want to use this, you might want to measure at frequent intervals, say maybe once every year or 2 years or every 6 months, and to see sort of what things you're doing, which are actually working for you. And I think that's the other beautiful part of this, is that this is really personalized medicine. You know, you and I both might take some type of peptide at baseline. My aging rate might go up, yours might go down, just due to our individual biochemistries. And so this is a really good tool just to see what's working for you and finally have something objectively that can be used to measure aging, rather than just trying to throw everything at it and hoping that one of those things sticks.
So I know some people are definitely thinking, you know, how is this different from a DNA test? Because a lot of people have been trained already to hear, oh, DNA testing, spit and tube, you do it once, you do your DNA test. That's it. Just once you have your data. This is really different. This is something you're not looking at. Tell us, you know, what are our genes, right? I don't know if that's included in the report, but that's not the objective.
Correct. Unfortunately, it's not included in the report. Although we are looking at some genetic data, it's very, very small amounts. And so generally what we're just looking at is that expression of certain locations in DNA profiles. And so we're not looking at what can happen. I always say we're looking at what is happening. What is the actual regulation of that particular gene? So that I think that it's a very, very different. And the good thing about these epigenetic reports is that they're changeable and modifiable. And so if you don't like your results, that's okay because you can change it. It is necessarily within your control, and you can make different interventions, which then would reverse that process or help improve that process. And so hopefully, and I think DNA can also be a little bit scary because you think, hey, is this my predisposition for life where there's nothing I can do about it? This is a little bit different because you do have some control. And we're really trying now to find out what those metrics are, I should say, that really move the needle in a positive or negative way.
Interesting. So a DNA report may say you have the breast cancer gene, but you would think that would be very useful for TruDiagnostics to say, okay, we know the rate of aging, how fast they're aging, they have all these things. And on top of it, they have a predisposition. Then wouldn't the test be even more accurate in terms of predictability?
Absolutely, definitely. And there, what you're getting to is what I mentioned earlier, where, as these things continue to improve, the best biomarkers will be composite biomarkers. They use information from many different organ systems, including that baseline DNA, as well as the epigenetic expression of that DNA. And then to go a step further, if you wanted to, we could even layer in, what mRNA is actually being transcribed from that epigenetic regulation. And then how that mRNA and RNA is be going into these peptides or proteins, which are actually being synthesized. And do they have any, variants in expression, or in the case of glycans, is it a post-translational process that's happening to those peptides or proteins? And then ultimately, are those peptides or proteins then metabolizing certain products in a different way than they normally would, which would give us that metabolomics sort of profile? And then ultimately, how does that lead to that phenome or the phenotypic process? How does that lead to changes in disease, changes in sort of the human health picture that we all see on a daily basis? And so all of that information is what we call this multiome. And that is exactly where this field is going, is the layering of many, many different points of information. We ourselves are almost done with a multi-omic trial, which will hopefully be the best age and death predictor ever published with Harvard. So we're putting a good bit of money, I should say, into this study to look at, a couple thousand patients with full multi-omic sequencing. The only thing we're not doing is that transcriptomic, that mRNA analysis, to sort of create this ultimate big composite picture of how well people age.
So when you say multi-omic, you meaning taking different biomarkers and putting them all together into one amazing thing.
Exactly. And
Okay.
generally doing that would be very, very expensive. If we ask everyone in the world to go get your proteomics, to get your, your untargeted metabolomics, all of your DNA sequencing and your epigenetics, that's really expensive. It's pretty much impossible for someone to do. I mean, per patient, we're spending a couple $1,000s for this study. And so what we need to do instead is to really have a way to train that information back to one thing that we can look at. And for us, that's always been methylation. So what we're training is all of these different platforms: metabolomics, proteomics, those phenomics, all back to epigenetic methylation, so that we can then predict those outcomes with a relatively low price and really efficient type of test, which is what we're doing already. And so we believe we can train most things to be predicted through epigenetic methylation, including, again, all those proteins, or if it's your VO2 max, or if it's, how tall you might even be, or when you might die. We think all of that can be predicted by epigenetic methylation. We just need to make those correlations. And to do that, we have to get a lot of data.
All right, guys, get your kit now. We need more data. um, so you've already seen a lot of people and a lot of data. And what are your reports or your studies showing that which interventions are working best to slow aging?
Yeah, so I always like to classify this in a couple different criteria and areas. The one that we know the most about are epidemiologic studies. And so this is where we would get, this marker on several thousand people who have basically participated in these biobanks, which means that their sample might have been even taken 40 years ago. But what we're able to do is to look at their behaviors, look at their associations, and then see what is increasing on average epigenetic age and what is decreasing epigenetic age on average. And so there is a lot that we know epidemiologically. Things like, for instance, smoking or high BMI, or those types of things are very, very well validated. And I hate to say it, but also the majority of what we find is actually very, very intuitive. It's probably all those things already that you need to be doing, right? Getting the proper amount of sleep, making sure that you're exercising, but maybe not too much exercise, right? Don't drink, don't smoke, making sure you're avoiding chronic diseases and viral infections, right? All of these things we already know probably impact your health in a better way, which is actually, it might be boring, but it's actually a really good thing, because what it is we're seeing that this is corroborating all of what we know already, making sure that all these things that we know are bad behaviors or linked behaviors are also associated with, better aging or worse aging outcomes. And so it might be a little bit intuitive, but it's very, very important that we're seeing that consistency. What we're starting to find now, and this really just started happening in 2019, was these interventional trials where we're actually looking at a baseline measurement for individuals, then adding a certain program or protocol, and then looking at an outcome. And so seeing how those things are changing even on a month by month basis. And so this is something that is very, very new. There have only been right around 10 interventional studies published to date. So the information is not super robust. Most of these studies have anywhere from, maybe 20 to 50 individuals. So still relatively low powered, but we're starting to see a lot of really good associations. And so things, for instance, like the Mediterranean diet, we know are very, very good at working. Things like DHEA or vitamin D, these now have multiple interventional styles published to sort of say everyone should be doing this.
Hmm. So I imagine a lot of people who are investing in TruDiagnostic or by getting this kit at home, it's not cheap, first of all, and you probably have a lot of people who are trying to optimize their health. Maybe I'm wrong, maybe it's you're seeing all kinds of stuff, but it would be interesting to know, are we talking about people who are generally healthy, like the biohacking community, and just want to optimize, or are you looking at really sick people? Out of all those 13,000 or whatever more people, what's the percentage of you got?
Definitely people who I would say are more in this biohacking community, more of the health optimizers rather than those who are currently sick. And that has good and bad implications. The first thing I would say is that almost no biobank, any university system that's getting this information, is using healthy individuals. Most of those are using people who come to hospitals and are already sick. And so one of the best things about our data set is that it is a very, very healthy population, but it's also skewed in other ways. Generally, as you mentioned, the testing can be a little bit expensive. And so it's relatively skewed to a more wealthy or affluent patient population. And so our data set isn't perfect, but we are starting to find out things, particularly from our population, which are those healthy people who are just looking to improve this. And one of the biggest reasons that is a good thing for us is because we can actually draw the line in differentiation between these aging biomarkers and disease-related biomarkers. So sometimes it's really hard because, faster aging can predispose you to worse diseases. But at the same time, diseases can actually accelerate your aging process. And so it's really sometimes hard to pull those things apart. But by looking at a healthy patient population, we can get a really good signal of what indeed is aging and separate that entirely from disease. And so yes, our population is definitely more of those health biohackers. And that's actually where I would say we're getting some of the most exciting information, right? Because we're able to look at, let's just take, NAD related therapies, for instance. A lot of our practitioners are using NAD related IVs, but we're able to compare that with those patients who are taking nicotinamide riboside or the nicotinamide mononucleotide as these oral precursors to see on these large scales, these several thousand patients, what on average is having the biggest change, or can we see any significant associations from those people who do different protocols? And so that data set is growing, and we're finding out a lot of really interesting patterns. I would say we're finding some supplements that generally have really significant changes. We're starting to see procedures that have really significant changes. So we're looking at everything from bariatric surgery to plasma exchange and young plasma transfers to senolytic cell therapies to hormone replacement therapy. And so we're looking across a lot of different things. Unfortunately, we don't have a lot of data back yet enough to publish. We are seeing some trends which would be definitely worth talking about.
This is what frustrates me in my gerontology studies, is that having more money, you're going to live longer and you'll just have more access to healthcare. You have better education. Yeah, it's very frustrating, because to me, it seems as though all these wonderful biohacking gadgets and things and we have are just not accessible to the people who need it the most. So my dream is to have it accessible to everyone. Why wouldn't do you think these kinds of testing, all these kits are prohibitively expensive for these types of populations? When do you think, or if at all, they will be more affordable for everyone?
Yeah, definitely. And that's, I would say we're right on the precipice of that. Actually, some changes. So we're going to be coming out with a test probably within the next few weeks, which will be a lower price point version of this test with our most important algorithm, which is this Dunedin pace. And we'll also probably add in telomeres, although we're not, I would say, as high on that telomere report, that the Dunedin pace of aging is a very, very good report, which we'll be able to have at a much lower price point, almost probably under half of what we're currently offering it for. And so we're really excited about that to make it more accessible. And actually that algorithm is a really good proof and concept of this. They've looked at this now in young individuals, even children under 10, and found that the event of polyvictimization, their socioeconomic status, their level of neighborhoods, socioeconomic disadvantage, these actually influence the aging rate immediately by the time we're even under 10 years of age, and affect outcomes at even age 45. We could actually predict a lot of those outcomes we saw at age 45 in that longitudinal cohort just from the information we saw epigenetically at age 3. And so we 100% agree that this is something that will actually affect epidemiology on a large scale. Even just 2 weeks ago, there was a article published in the Lancet, which basically recommended that we don't use chronological age anymore. We use these biological age metrics to figure out how federal funding should go to different communities. So we're starting to see that transition because we know how important all of those factors are to this biological aging. And I also should say all of these factors also have signals in the methylome, as we call it. So all these methylation signatures we're looking at can actually be tracked back to certain behaviors or traits. And so that's how we're able to, for instance, see how much you've smoked across an entire lifetime, because there's a particular location in your DNA which will change based on your smoking behavior, which we've now identified and been able to control enough to be able to predict things like how many smoking pack years you've smoked across your entire life, or how recently you quit smoking. And we can do that with a lot of different behaviors, everything from air pollution to from the area you live, to sort of how much you might be drinking from hydration. And so if there's one big takeaway from anyone who's listening to this, it is that epigenetic methylation as a biomarker is one of the fastest growing areas in medicine, and will impact every area of medicine over the next 10 years. And in the next 20 years, they even anticipate to overtake blood testing. This is a huge area of medicine that will affect everywhere. But right now, the thing that's been taking the lead has definitely been these aging diagnostics. And that's where we hope to push it forward, really, really far.
That is so exciting. It's personalized medicine, really. I think it's wonderful that we have that. And I want to know, because this is a podcast for women over 50, what are you seeing in your data with these women, and what kind of interventions do they need to take, if at all?
Yeah, there are a couple interesting, really trends for women that we see, which I would say are really, really unfortunate. When we talk about these trends, women, they don't get too excited about them. But one of the things that we know, unfortunately, makes biological age worse is the number of pregnancies that you have. Pregnancy is a stressful process. Sometimes these will cause relatively transient increases in biological age. So there's a really good study that was just published a few weeks ago that shows that things like pregnancy or even surgeries, anything that has these sort of short periods of stress, can cause transient increases in biological age. But even as a whole, actually, generally the number of sort of gravida or number of pregnancies that these women have will increase their biological age. And so if you're having more children, this is definitely another metric we might recommend you take, right? To see where you're at, and then to, again, see what works for you to get that lower. So unfortunately, that's a trend. The other trend that we see is that women generally are better than men. So that's the good news. Almost across the board, almost every woman of the same age group will do better than men of the same age group. Men just have worse aging. And we're not really sure why that is yet, but we're definitely trying to uncover some of those links. So that's the good news. The unfortunate other bad news is that menopause is one of those areas where this is going to be markedly increased. So as you start to become perimenopause or come to menopause, specifically, your aging rates start to really accelerate. And you can see that, for instance, in things like our rate of aging metric, but even in these overall biological ages. And so in menopause, it is an important transition time. We do know things such as hormone replacement therapy can actually slow that rate. Not that I would say, are necessarily advocating for hormone replacement therapy yet, but we do know that it can definitely slow those aging rates, and maybe even slow some of those symptom developments as well. And there are actually algorithms in the works for women to look at these epigenetic markers to tell you how much total lifetime estrogen you've been exposed to, which might be helpful in things like breast breast cancer, or there are even algorithms that can tell you how likely you are to have some of those menopausal symptoms, like hot flashes, for instance. And so, and so these biological age markers also relate to a lot of just generally high correlations to women health outcomes, like, for instance, breast or cervical cancer, or even menopause, for instance. And so there are a lot of those correlations and links. But the good news is that women is that they're generally doing a lot better than men. But there are also several points in life where you might want to pay special attention to this.
Super interesting. So you're saying the more pregnancies you have, then the faster rate of aging. So I thought, but are you sure it's due to the pregnancy? Because I imagine somebody with 4 or 5 kids, I mean, it's not the pregnancy itself, it's just the lifestyle that you have with trying to manage them.
Yeah, that might be a factor as well. Unfortunately, right now we probably don't have enough data to tie those two apart. I think that it might be good to look at mothers who maybe give up their children for adoption to draw that link, but that's a really hard subset of individuals to test, and so we don't have that data yet. But it is looking like, just due to the increases we see transiently with pregnancy, that it's probably something biologically rather than just the correlation to maybe some of those lifestyle factors as well.
Mhm. Okay. Hold on one second. I hear too much background noise. Hang on.
No problem.
Okay. Sorry about that. Okay. So women over 50, we need to, it would be very interesting if we could test ourselves before menopause, if you're, or perimenopause, I guess, and then test again just to see the rate of aging. Do you see any people doing that?
So unfortunately, this data hasn't been done longitudinally. Most of the data that exists for that type of investigation has been mainly from these large epidemiological studies, where we're sort of taking individuals at several different time points. We're not being able to see longitudinally how menopause is affecting, but we can see those trends from maybe people under 50 or over 50, for instance. And so with that being said, we are seeing that. That's where we're seeing that age acceleration. But as different cohorts begin to age, like that Dunedin pace of aging marker. They had that cohort that started in 1972. We're still following up with them now. And actually, our next follow up is when they're 51. So we'll actually be breaking that 50-year age mark here relatively soon. And so hopefully things like that cohort will be able to tell us a little bit more about how some of those sort of, sex hormone related conditions are affected by, biological aging and vice versa.
Do you see a lot? I mean, a lot of the biggest difference that you see between men and women, or just that women are aging slower up until the menopause point, and then they go pretty fast. Any other differences other than the pregnancy and the things that you mentioned?
Yeah, so at the moment, I would say there aren't a lot of really large differences in the cohort, other than men tend to have more accelerating across the board. That includes
Mhm.
even after 50, and when women are going through menopause. Women might tend to accelerate their aging process after menopause, but they're still better than men who are aging
Mhm.
rapidly generally their entire lives. And so we are seeing, I would say, just that is the main trend. I wouldn't say there's a lot of differences we can really tie up for men and women just yet. But we're starting to get there, I would say, with a lot of this other demographic information. So, even a lot of the questions that we ask on the intake are actually, we're used to look at correlations, where we're saying, if this person is taking, for instance, NAD, and how does their aging rate after 50, change? Or we could look at that, for instance. And so we're using that data in a lot of different ways internally to be able to look at those correlations. But right now, I would say there's not a whole lot of differences between men and women in the aging process.
What about COVID? Because we saw, can we talk about COVID? I don't know. You can say the C word, but there, because you started the company right at the beginning of this whole pandemic, and you've probably seen a lot of data of people who've had COVID, who haven't had COVID, who've been vaccinated, not vaccinated. Could we talk about that? What you're seeing?
Yeah, definitely. And we actually just published a study with Cornell and Yale on this exact topic in Frontiers, where we actually looked at longitudinal patients who had a baseline study with us and then had developed COVID, and then we tested them again. We did the same actually with the mRNA-based vaccines. Those who haven't gotten COVID, who had a baseline measurement, had been taken, a Pfizer or Moderna vaccine, and then had sort of a measured outcome. And so we just published this data, and we actually were the first to use sort of a new clock on this called the principal component clock, which is incredibly accurate clock developed at Yale by Morgan Levine and Albert Higgins Chan. And so this clock is really, really important because one of the problems with these epigenetic clocks previously has been their level of precision. So I think that, you ask if I didn't answer those survey questions and send in the same sample, how would those differ? And we also asked that question internally. If we took the exact same sample and measured it twice, how similar are those results? And typically this has been one of the biggest problems with epigenetic age, because some of the first clocks had mean absolute errors of over 3.9 years, right? Where the same sample could have a wide variation. And that can be really, really difficult to make into a personalized medicine journey, because if you're testing within 4 years, how do you know if it's noise in the metric or if it's an actual age related biological signal? And so that's been a huge issue. One that we were the first, I would say, commercial company to outroll those PC clocks. And we were the first to publish on them with our collaborators at Yale and Cornell. And so I want to just take a moment to say that those really accurate clocks, so those principal component clocks, are really all that we use now. And so you can be very rest assured if you're testing with us, those results are going to be very, very precise as well as accurate, meaning that they're going to be predictive of disease. But with that being said, those clocks were really important for COVID because when we used the regular clocks, we didn't find any association with COVID or the mRNA-based vaccines. Our population sizes weren't big enough. But when we switched to these PC clocks, we saw a lot of significant correlation. And what we saw generally was that people who had COVID and were under 50 years of age actually had a slight improvement in their biological age. Vice versa, though, people who were over 50 chronologically tended to have sort of a worsening biological age if they got COVID. So that told us some really interesting things, and generally allowed us to speculate on why we might see that difference. And we believe that the reason we see that difference is that people under 50 had the immune system, which was able to mount an effective response to the virus. And then as a result, improve, sort of conquer the virus and then improve itself. Whereas those people over 50 maybe didn't mount an immune response, which was as positive. And then were unable to handle all of that sort of inflammatory burden or all those negative things we see as the sequelae of COVID-19. And so it was a very interesting thing, because the split that we saw was not in sort of an arbitrary split. It was very, very clear, and definitely saw that maybe the immune system was a reason we might have improvements or decreases in that biological aging process.
Hmm. And these are people who are generally healthy, right? I mean, this is not
Correct.
your standard American diet type of person. So very interesting. And so, yeah, what about with the vaccinations? Have you been able to see any differences with those populations?
Yeah, we have. And again, we had significance in that population as well, where we tested, I think, 46 people who had a baseline, then the vaccine. And we did not separate Moderna or Pfizer. We didn't want to get political and say, which one you should test. But we looked at them as a whole and saw very similar trends across those, regardless of brand. We saw that most people had actually improvements in their aging rates. And so most people improve with mRNA-based vaccines. And this, is something that is a little bit controversial, but I should also mention that it's a low-powered sample size, and we actually believe that probably the reason that we saw that change was not necessarily because that we were seeing improvements with mRNA-based vaccines, but just because we were seeing maybe that immune system be stimulated and increased, and that improvement might just be sort of a residual from the changes in those immune cell subsets that we're measuring. And so we're not really sure why we saw it, but we did. The next study that really needs to be done is, does any type of vaccine maybe positively increase the epigenetic age for a short duration, a short period of time? And so we're hoping to do that with, for instance, things like the influenza vaccine, all right, your traditional flu vaccine, to see if we see similar changes. And we think that we probably will, and that there might be some sort of just internal signal whenever you're encountering a virus, which improves the immune system. It creates a sort of adaptogenic response, this hormetic response, which then helps your body sort of regulate itself in a positive way.
Hmm. Very interesting. So would somebody who's interested in testing themselves, then if they just overcome an illness, whether it's COVID or something else, or just took a vaccination of some kind, would you recommend them to wait for a while until they test?
So we actually don't. And the reason being is that we hopefully can control for those things. I mentioned that one of the things we're able to do in our reporting is to tell you what percentage of what type of immune cell you have. And that's really, really important because if you have for instance, a virus, you might increase your natural killer cells or your B cells, which then might throw off this testing. However, the thing that we're able to do is actually control for those immune cell changes because we know how many cells you have. We're actually able to say, have algorithm tweaks to make sure that we're controlling for what's in your immune system. And so hopefully cell percentage or proportion won't affect these scores at all. And so we don't necessarily recommend people, having to wait specifically after a virus. But with that being said, there might be some rationale in saying that there are signals that maybe for viruses we don't have as much data on that we can't quite, predict those signals or control for as well. And so I can understand if you would want to, but by definition, you don't necessarily have to.
Mm. What blew my mind was when I was trying to draw my blood and I could only get you a little drop. You still managed to get my data. I mean, that's incredible. Do you have did it arrive? Do you have any of my data in front of you?
So I'm sorry, Zora. I'm trying to wait. Unfortunately, it's still a little early here in the office, and I haven't gotten anything sent to me just yet. But I know that we have it coming, and hopefully we'll be able to talk about that very, very shortly.
Okay.
So I'm hoping to expect it any second now.
Well, you just interrupt it if it comes in. Then this is data. This is very personal data. How can you guarantee that it's going to be safe and sound?
Yeah, so we actually bought our building for really the data storage that existed here, their own private servers and the security. But it actually for us now, we actually use all Amazon Web Services because the security and encryption is just so much better than anything we could do on our own. And so we take data security very, very seriously. We're SOC 2 compliant, which is basically a certification which makes sure data security is prioritized. We keep everything de-identified from your name, and so only under very, very rare circumstances when we're maybe doing these report reviews would we ever link the two data points, and even then it's not linked in our system. It's just sort of linked so we're able to use intermediates to search. And so the other thing we do is if you ever want us to destroy your data, you can easily just send us an email and we will absolutely get rid of it, which we do actually fairly frequently for a lot of our customers. I would say right around 10% of everyone we work with ask us to delete their data right upon review. And so we're able to do that. And one of the problems with that is we're not able to get all the updates that come across. And so it is definitely a personal decision, which, but we do everything we can in order to keep that data security. We also have a very, I would say, adamant policy about sharing that data. We do not share with anyone externally without a completely authorized private consent. And even then, we generally like to, whenever we work with universities, interpret the data on our end and share that de-identified large metadata with anyone as well. And so we believe the data is yours and that we should not be monetizing it.
Hmm. That's good to know. So before I open up the questions to our guests who are here, can I ask how old are you? Because you look so young, like to be talking all about all this, but you're obviously super intelligent.
So biologically or chronologically is what I ask now. But actually, whenever I first started this company, I was 29 years of age. Now I'm 31. And whenever I tested at 29, I actually was right around 31.5 in my biological age. And so, being able to test myself fairly frequently, and my blood is actually the standard that we use almost on every run. So I'm able to test myself really, really frequently. And the knowledge that I've gained over, I think, those 2 years has allowed me to actually reverse my biological age by right around 2.5 years. So actually now I'm the opposite, where I was originally 31.5. Now I'm probably 31.5 chronologically, but right around 29 in my biological age. So over the course of 2 years, I sort of flipped that entirely, which I'm very, very happy about. One statistic I always like to mention for people is that if everyone in the world were 7 years younger biologically than they are chronologically, we would cut disease in half. So 50% of people would no longer be sick, and you'd be decreasing your own risk factors for mortality by 50% as well. So that 7-year age gap is absolutely our marker for everyone to try and hit. That's what we really try and encourage people to do.
7 years. That's quite amazing. I think we all need to shoot for that. It is quite surprising. You've been able to reverse it starting a new company. I mean, it's really stressful what you do.
Well, yeah. Surprisingly, that is, I think, one of the biggest things to change is stress. Stress has a major impact. When I first started this company, I was not a big proponent of mindfulness or meditation, or I always sort of was thinking, sitting there, am I doing this right? Is this meditation? Is this mindfulness? Is this relaxing? And sure enough, I think that one trend we've seen is that people who incorporate those tend to reverse their aging process. And even with we think that a lot of that link is mitigated through stress hormones like cortisol, for instance. In the original sort of 2013 Horvath algorithm, they look at 353 locations on your genome. And of those, 85 of those are located at or near glucocorticoid receptors. And so we're starting to see the importance of some of those things like stress management. And if there's one thing I tell everyone that generally has a positive effect on our cohorts, it is absolutely stress management. And it is things like DHEA, which can limit the effects of cortisol and drive that down a little bit as well. So I'm a big proponent of anything that like that, even though whenever I first started, I absolutely was not.
Mm-hmm. So you're now meditating more? What's your hack?
Yeah, so I would say I've been doing a good bit of yoga, which for me is a way I think to feel like to have I'd say pace meditation, something that I can follow, and I don't have to always be worried. Is this that outcome that I'm supposed to be achieving? I just sort of get involved in the process, and to me that has worked. And I would never did that ahead of time, but I would really credit that to being probably one of the biggest things that changed my score. Although I'm the quintessential self biohacker, try a lot of different things as well.
Great. All right. We'll have to have another podcast for those secrets. So I would like to ask anyone who's here, if you have questions, please unmute. I know I'd love to start with Magdalena because I know she's always got great ones. Magdalena, unmute. Yeah.
Is it okay now?
Mm-hmm.
Yeah. Actually, Zora, you have asked something I wanted to as well, and that was correlation if taking this test, but you have had COVID, let me say, some time ago, because I got COVID in March and now did a test, because unfortunately last year I bought it, but it was lost somewhere, my blood sample. We were waiting for 3 months and then nothing happened. So you resend it. Thank you so much. So I did it after getting COVID, and I was really very, very sad because of this, because now I don't know the results, which there will be in the nearest future. It might be worse because of COVID, or maybe not. But if I did COVID test, I did it last year, and if you did the result, maybe the results would be better. So we'll see. I will wait. So I wanted to ask the same, but if I understood right way, it doesn't matter, right?
So I should say there is damage that is done to the body by a COVID infection, which is a real biological age impact. And so, for instance, I always talk about telomere length as one of those things. Now, in multiple studies, not just looking at epigenetics, but have actually been looking at telomere length, we see associations with telomere length absolutely being lower after COVID infection, and actually having some relationship to maybe the severity of the infection as well. And so there are some things which will absolutely have a biological impact, which are a real biological impact of that aging and disease. Unfortunately, with our study that we looked at with COVID, we haven't followed up with those same individuals a year after they've taken that metric to sort of see if they can recoup from some of those, damage that we are seeing, and especially in people over 50. And so I would say that if we are seeing a signal, it is, and especially several months after COVID, it might be a lasting or I would say signal which is probably sort of something we want to overcome. And so it might make it worse, but it doesn't necessarily mean that it can't be changed back.
Yeah, I believe this. Actually, I was waiting for 2 months just because of this matter. I hope it will be helpful. We will see. Otherwise, I wanted to ask you, do you know maybe to compare your kit with the kit of Zymo Research and the kit of Life Length? Because these are two kits I've done the blood tests a few years ago, and my results with telomere life length test was really bad. I was like 8 years older biologically than chronologically, and I couldn't believe it. And then I did it 1 year later, the Zymo Research methylation test, and my result was actually pretty well, 4 or 5 years younger than my chronological age. So now I'm really so happy to get your results, because I would like to compare different companies, maybe different tests. What do you say?
Yeah, I'd love to always talk about specifics. So in the case of life length, they're using qualitative PCR to measure the actual telomere length. And so that is actually probably going to be more accurate than our telomere length testing at being able to tell you the length of your telomeres. But to counter that, I would also mention that our measurements of telomere length are more predictive of disease outcomes and have double the correlation to age as traditional telomere length testing. So I would actually think that these methylation markers estimation of telomere length is better than telomere length testing. However, if I was going to do actual telomere length testing, I'd probably use sort of, instead of using qualitative PCR, use fluorescence in situ hybridization testing for telomere length, which are done by a couple labs here in the United States. Again, though, I think that multiple studies have shown that telomere length, although correlated to age, is not necessarily predictive of these outcomes, which is why I think it's not as important. Another study recently looked at twins from Stanford, and it looked at the phenotypic differences in aging of those twins, right? So they have the same DNA. A lot of them have grown up in the same environments. And so it was trying to basically say, out of all the changes we see from aging, which is due to what? And they found right around 2% of that is due to telomere length, while right around 30% of that is due to these epigenetic clocks. And so I think that sort of shows you why we would prioritize these epigenetic clocks over things like directly telomere length. And so they're both important processes in aging. If you immortalize telomere length, you'd still see epigenetic aging. And if you reset those epigenetic clocks, you'd still see telomere length attrition. And so two very separate processes in aging, but at the same time, both are, if we're looking at which one's more important, it's going to be those epigenetic clocks. And so I guess that brings me to the Zymo. And so Zymo actually was the first company I ever toured when thinking about setting up this company. But unfortunately, I thought that we needed to create our own, because Zymo is doing two things which I think are not optimal. One is that they're using, first off, the 2013 Horvath clock, or a variant of that they've created. And that is a first generation chronologically age trained clock. So as I mentioned earlier, that's one that's been trained to predict the chronological age of a patient. So the better that one gets, the closer it is to getting to something that you can just ask for, right? Your biologic or your chronologic age. And now these second and third generation clocks are all that I would really recommend using in that field. The other thing is that the amount of data they're looking at for you. So they're really only looking at right around 1,200 data points, whereas we are looking at over 900,000. So significantly more, which is able to tell us a lot more, and also have much higher accuracy. As I mentioned, those PC clocks, which can take sample sizes needed from 100 to 5 patients. Those use 80,000 CPGs, which is significantly higher than the data that Zymo would be collecting. And so they might give you better scores. They might tell you that you're younger than we would, but at the same time, that doesn't necessarily mean they're better or they're more accurate. And so in order to do that, you have to look at those data, the precision and the hazard ratios, and our data would outperform those on any category.
I would also suggest something, to have a remark regarding these blood plastic tubes. I saw that Zora did it without a problem, but me and my partner, we couldn't. Every time we went to the lab to take our blood and, to really fulfill much more these milliliters or whatever, how to say, blood to this plastic tube, because what is in the kit, I think I cannot really take enough of the blood. I don't know now, you said before that, of course, it's not just 5 or 6 days. It can last longer, this blood, but I'm living in Slovenia, and I guess it takes a little bit longer to come to your place. So I definitely think that this kit is not really, not optimized how to take a blood, because we couldn't get the blood at all from our finger with this kit. So every time we need a time, to go to the lab to do this, and to have a little cost, they took the blood and so on. So here is my suggestion, maybe to have an idea to change something in the kit.
Yeah, so we already have, and so whenever we first rolled out, the only reliable method that we had certified was those EDTA tubes, and so that was definitely not the easiest. To put the blood from your finger into that tube is sounds like an easy process, but in actuality, it is rather difficult thing to do. You get blood all over the place. It's really hard to concentrate it, and so for us, we've always known that that's not going to be a lasting solution, and so we've been really trying to validate this different types of collection collection methods. And that's why we finally switched over to these blood spot collection methods. And it makes it a lot easier because the capillary action of that dry blood spot card actually is able to wick away the blood from your finger very, very easily. And so it allows us to sort of just, prick the finger and then just sort of dab it a couple of times in order to collect that blood that's needed. It's a much easier process. We also have, at least in the United States, some automated collection methods where you can just sort of put almost like a continuous glucose monitor on your arm. You press one button and then
Yeah,
slowly the
exactly.
blood
It's so
will
easy
come
and
over. Yeah,
you
and those are
don't
called
feel it at all. Yeah.
yeah, those are called the Tasso kits. And so in the United States, they're available. Unfortunately, they haven't been licensed in several other countries yet. And so we can't ship them internationally. But the other thing we like about the blood spot card is it makes sure that it gets custom clearance a lot quicker, which is sometimes one of the big things that we've had with international clients and customers is that unfortunately, if your biological specimens in the age of COVID do not go through customs very easily.
In our country is prohibited. I shouldn't even tell, to UPS in Slovenia that it's actually a blood sample. So I said, just it's something for you, I didn't want to explain because I'm not sure that this could go through,
Exactly. And so now with those blood spot cards, it passes customs clearance very, very easily because they can do what they call a bin test, which means that they can bin the envelope. It can go through without additional inspection. And so now that method is blood spot, I think will be something we'll stick with a long, long time. And so hopefully the next test that you do will be much easier to collect.
This is really cool. And now the last question regarding the price, you were explaining a little bit before. We all find it very expensive because it's not just your test, it's other tests, Dutch test, this test, that test, glycanage maybe to try to compare and so on. And all tests are very, very expensive. So I'm following Dr. Sinclair as well. So I guess that you might know that the brand he's doing now
Tally,
for measuring biological age, I think the name is Telehealth or something like that.
Yeah.
And he said that this is now in the nearest future coming, and it will cost only a few dollars comparing to your test, which are or more $100. So what do you say? How will you and other companies prepare to be, let me say, that you can compare with the others,
Yeah, definitely. So
people will buy something what is much more cheap. So this is really an important question because I guess that many people would like to do this test, but it is just too expensive. So if this will be few dollars only, then everybody can try to do it, not only biohackers, because we biohackers, I think on our list is first supplements and tests and devices, and then the second nothing, We are spending a lot of money for this, but we are biohackers and we are following all these technologies, all these researches and so on. But let me say in brackets, ordinary people who don't know much about it, they don't care if they buy or not, but it would be important for everybody, especially 50 plus age to test. And I loved what Dr. Sinclair said, that actually every doctor should give this test or this, I think it's a kind of bio button to the customer, to the client, to test what's going on with his health. And I think this is really important for the future.
Definitely. And I can completely agree. Getting the price down is absolutely something that needs to happen. But I would say that it should not be done at compromising the validity or accuracy of the test. And so I think that's an important thing to note because, again, there are a lot of biological age tests out there, and you can use anything from, one that uses blood-based biomarkers to one that uses telomere length. But we will consistently provide the best biological age test on the market. That's what we want to do. And so in the case of Dr. Sinclair and Tally, we work with Dr. Sinclair. We're doing a couple studies with them at the moment. But Tally will be using a method called TimeSeq, which is a new sort of method developed at Harvard, which is able to drive down the cost of profiling individual sites for epigenetic analysis. And so in the study, for instance, they say that you can get down to as low as a cost of goods of $4 per sample, which is not necessarily I would say, completely transparent to all the things that go on. And so at $4
Most
per sample,
accurate, I guess.
yeah, so you can look at a couple locations off of an already defined clock. But as I mentioned, with those those PC clocks, which I consider the standard of the industry due to their accuracy, right? You don't, whenever you're measuring these things, if you send in 2 samples, you don't want them to differ by 4 years, right? You want them to be as accurate as possible. In order to do that, you need several thousand CpGs. And that is still not possible, even with the time-seq method, to do it at really low cost. And so at the moment, the most efficient method is still that Epic 850K array, which gives you the most data with a relatively still low cost point where you're not having to do full genome bisulfate. And so, and so with that being said, TallyHealth will be coming out with a method that will be a chronologically age trained clock. And again, I think that's a little bit of a regressive step backwards, because you don't want a chronologically age trained clock. You want a biologically age trained clock, because it's capturing that biological signal, and then you want it to be precise. And so we are not going to compromise accuracy or I would say precision as a result of doing these metrics, which is why one of the reasons why our lower price point test, which will be right around that $200 range, will be that Dunedin pace of aging, because it is still very, very, very accurate. We measure that accuracy via something called the intra-class correlation value. And that is saying we take the exact same sample and split it into 3 or 4 tests, how correlated are all those tests to each other? And generally what you want to see there is an ICC value above 0.9. If you don't have that ICC value, then you're definitely losing accuracy, and you're getting so much noise that you won't be able to effectively see what's aging and what's not. And so that Dunedin pace of aging marker is really, really good at making sure that it's accurate. The other thing we talk about is accuracy. And so this is, again, been an issue with these biological age trained clocks, because if you look at the first clocks, like for instance, the Horvath clock from 2013 that Simon was using, or the Hannum clock, or even PhenoAge by Morgan Levine when she was at UCLA, all these clocks are trained to chronological age. And whenever we use them in this calorie study, which is this basically 2-year, 11% caloric restriction study, caloric restriction is one of the most well-validated therapies for anti-aging and longevity that we know of. It works in almost every animal species and has been studied several times to have life extension effects in humans. And so, unfortunately, when using those first generation clocks, those actually go up with caloric restriction. So we actually see increases in biological age whenever we're seeing caloric restriction, whereas the newer generation clocks like GrimAge or the Dunedin pace of aging, they go down significantly. And so these first generation clocks that are trained for chronological age in 2013, they were the best thing that we had, but now we know that they're not. And so you need these biologically age trained clocks, and eventually time-seq will be available to be used in some of these newer clocks, but at the moment it's not there. And so we'll continue using that array method, which gives us the most precise, the most accurate, the most responsive to biological interventions, and gets us a lot more data. And unfortunately, we'll have that lower price point test for the algorithms which are ready for that, like the Dunedin. But until that happens for all of the algorithms, we probably won't be able to lower the price that significantly. But I would say within 2 years, you'll probably see a lot of these metrics under $200 in cost.
Yeah, most probably. Thank you so much for everything.
Yeah, my pleasure. Thanks so much.
Great questions. Appreciate that. So yes, I'm glad that someone's on the track, and you included in trying to get a price point for people who really, really need it. Claudia, do we have time for one more last question, Ryan?
Yeah, absolutely.
Okay, Claudia, any thoughts or questions? You can nod if you know, or yes if you're too shy, or unmute. Oh, wait, you have to unmute. Okay, you should be. Can you hear Claudia? I can't hear her. Her audio is unmuted, but I don't hear anything. Okay. All right, Claudia. Unless you figure it out. Yeah, we can't hear you. We can't hear you. You can keep trying. So, okay. Brian, I am so grateful for your time. Really, it's been amazing. I've learned so much. I want to just let people know, if you want to get tested, they can go to TruDiagnostic, and that's TRU without the E, Diagnostic, and they can find out so much about you and the company and what this all encompasses. Then on Instagram and Twitter, you got a YouTube channel, Facebook, LinkedIn, it's all there. I'll have all the links in the show notes. And you can get $50 off with the code ZORA, ZORA, and get your kit. And I would love to know how your experience goes if you do this. Feedback is super important to me. And I would love to ask Ryan if you have any final words before I let you go.
Yeah, I just wanted to say thanks so much, Sora, for having me on. Hopefully, we'll be getting your results anytime soon and be able to go over those specifically as an example to anyone who's listening. But thanks so much for having me, and hopefully we'll talk again soon.
Wonderful. Have a good day, good night, good morning, wherever you're at, and we'll see you next time.